Very nice to meet you. I'm very pleased to see you in person.
Likewise, it's a pleasure and honor to meet you in person.
Call me Tasku. That's easy for you. Tasku.
Tasku.
Yeah. Okay. That's perfect.
And you can call me Ugur. Is that possible?
Ugur, yeah. Ugur.
Yes.
Okay. First of all, I'd like to express my sincere appreciation your contribution to the world, and also personally congratulate your great success for generating and offering the RNA vaccine against the COVID-19. Thank you very much.
Thank you, Tasco. This is a great pleasure and privilege to be able to help.
So I'm very much curious the process you came. The first I'd like to ask you a little bit of the personal history. I read many stories. You studied as a medical doctor and wanted to do the cancer RNA vaccine. So could you briefly describe what you wanted?
Yes. So from my background, I am tumor immunologist, and I'm also a doctor. I have seen patients. It was in the 80s when we did not have this great immunotherapies to offer, but only chemotherapy, and patients were dying. And as a tumor immunologist, I was really interested to develop, to activate our immune system to attack cancer cells. And our early research showed that tumor cells have tumor antigens. A number of so at that time the first mutated tumor antigens were discovered, but also this cancer germline genes like MAGE, GAGE, and we discovered an approach which allowed us to identify tumor antigens. But we understood that every patient has different tumor antigens, and we said, okay, we need really a cancer vaccine approach that could allow us to make vaccines for every cancer patient. And we tested different vaccine approaches, viral vectors, peptides, recombinant proteins, DNA vaccines, and then realized that mRNA vaccines could in principle, from the concept, be useful to go for.
Right. So to make use of RNA as a source of antigen is a very good idea, but I'm pretty sure you face the problem as RNA just single injection into cell. We also experience this cause a lot of problem.
Yes, it was the early time of the mRNA vaccine research. There were a few groups worldwide. This was end of the 90s, and we realized that mRNA, you can transfect dendritic cells in vitro, you can stimulate T cells, but in vivo this was a weak approach. And we understood that we have to change the mRNA. We have to do something to make it more potent. And by this we started a research to understand what makes an mRNA stable. How can we get the mRNA into the cells? How can we benefit from the immune activity of the mRNA? This was my early research.
Right. So that's the very important critical point. And I also read now your colleague Kariko also came to that question, and certainly you can tell me how you get together and how this big hurdle was overcome.
Yes, so Katalin Kariko did her research in University of Pennsylvania.
Mm.
She was publishing her research. We were publishing our research. Yeah. And at a certain time point, we understood that bringing these things together could be really helpful. Yeah.
Right.
Because we had developed extremely potent tools to stimulate the immune system. She found a way to make the mRNA less inflammatory.
Right.
Yeah.
Right.
And so we brought these concepts together. Yeah. And came up with a vaccine format which is suitable, and tolerated in a better way at higher doses. So we have at the moment different types of mRNA for the cancer patients where we need really the strong T cells. We rely on a different mRNA, not on the nucleoside modified mRNA, but the iodine mRNA, which provides strong TLR stimulation.
Right.
That means activation of the immune system. That's what we need in cancer patients. For the infectious disease, we used the nucleoside modified mRNA, but developed formulations and improved the targeting of the mRNA to dendritic cells.
Right.
So dendritic cells are the most important immune stimulators in the body. And we found a way to reduce the dose for our vaccine. So the challenge, of course, is if you need a too high dose, then you waste mRNA. You end up with a situation which is not anymore well tolerated. So the challenge was really to make the vaccine, in combining everything what was published and what we had learned, to come up with the most potent molecule.
Yeah, I read your paper, not in depth, but I realized you incorporated many aspects of the modern RNA biology and to make that most efficient way.
Yes. Yes, it is the wonderful aspect. So people often ask the question, what is basic research, what is applied research?
Right.
Yeah? And Louis Pasteur said this is the same. Yeah, because it's just one research. There's only one research, and the application are the fruits of the research. Yeah, and we needed to learn how mRNA is controlled in our body, the natural mRNA. Yeah,
right.
We had to understand the rules, and what we had also to learn is how viruses bypass the control systems of our body.
Right.
And by learning from nature, we could use the tricks of nature, and bring it back into our mRNA. And since an mRNA is a really big molecule with different parts, you can imagine that this is really many bits and pieces which decide about the fate of the mRNA. And we independently optimized these bits and pieces.
Right.
Yeah. And then we tested these bits and pieces together and learned if they add up, if they are synergistic, and came up with a optimized molecule which has really different pieces to come up with a close to perfect molecule for our vaccine.
Right. I understand you modified 5' 3' UTR and codon change, all kind of the nice technology combined together. So that's very nice. Yeah. You said the application and the basic research is not such a big barrier sometimes. Probably I didn't study cancer when I worked on the PD-1. I was totally in a different field. Like actually, I'm studying how vaccine can activate immune system. This basic question has been my lifetime project. But suddenly I hit the PD-1, which regulate immune system in a negative way. which itself is rather unexpected, and then got into the cancer programs. And I understand we meet in the similar field, cancer immunotherapy.
Yeah, absolutely. It's such a serendipity. Yeah. And that means you need to be prepared, yeah, not only to achieve your goal, but to be open to see things as they are, yeah. And it's very similar with us. I am not infectious disease vaccine expert, yeah, and I am an immunologist trying to activate our immune cells, CD4 cells, CD8 T cells, antibody-producing cells. And at a certain time point, we realized that what we did for cancer could be useful to develop an infectious disease vaccine.
Yeah, so from the cancer, you switched to the infectious disease. So what made you this quick snapshot change?
Yeah, so our cancer program, our cancer, the goal that we want to accomplish in our cancer program is to get a genetic sequence of the tumor of the patient, so the information about all mutations, and then make a tailored vaccine and deliver the tailored vaccine to the patient who has the cancer. And of course, about making a vaccine, genetically engineered vaccine, in the fastest possible way. So that means we had set up our laboratory and the methods to be as fast as possible.
Right.
We had spent 20 years to optimize our mRNA to be expressed in dendritic cells to stimulate immune responses.
Right.
And we understood when we realized that this outbreak in Wuhan will most likely become a pandemic, we understood that having the ability to make a potent vaccine in the shortest possible time, that is what we did for cancer patients, could be useful, yeah, and we could be among the fastest groups coming up with a vaccine. So we really pivoted the company, yeah, our research, and said we have to develop a vaccine. It's an obligation. We have to try to help, yeah. And the problem was there was so little known about this virus, yeah. And we could not just make one vaccine and hope that this is going to work. And our strategy was to start with 20 candidates, which were chosen in a rational way.
And how many of them will turn out to be the good one out of 20?
Yeah, so it was really an exciting story, Tasku. It was, we started with 20 candidates. In the animal experiments, we could reduce that to 4 candidates. Yeah, and when we went into the clinical trial, we realized that 2 candidates have very strong, were well tolerated and had very strong immune responses, and we were able just a few days before the final decision for our phase 3 candidates to get all the data, the immune response data, the antibody data, the CD4 T cell data, the CD8 T cell data, and it was very clear. So this is the candidate to go. It was BNT162b2.
That's wonderful, Ugar. So one simple question. You had a very strong competitor, Moderna, and I understand you were very close, but you did first. What make you this you think the difference?
We called our project Lightspeed, yeah, and the idea behind this name was really to go the fastest possible way that physics allows us. really rethink everything what we do. Yeah,
I see.
And so we implemented 24/7 program in our company. So teams were coming when other teams were going. Yeah?
I see.
And our teams worked on the weekends. Yeah,
I see.
And this really made a difference because this was such a commitment. Yeah,
Right.
And it reminded me back to my PhD time where I was every day in the lab. Yeah, and this is back to the roots, the determination and asking the question how we can come up with results.
Wonderful. And the effort was now you got the products and fruit. So
yes,
yeah, congratulations again.
Thank you.
So I understand your team published before everything is approved. So what made you feel this kind of the open information exchange is important?
It is really the idea of the project Lightspeed. So it is also about human nature. Yeah, we know that in a well-functioning group, transparency is key, trust is key. Yeah, and being avoiding any type of ego is key. Yeah, so we really counted on the most important values of mankind, on collaboration, on trusting, on synergy, yeah, and transparency. And I really like this and this publishing preprint publishing, yeah, which gives us the opportunity to see what others are doing, to learn, and of course being careful and not to take everything granted because it's not peer reviewed, but it gives really the opportunity to share information and to learn.
Good. So sometimes, scientific community, this publication causes lots of stress. And if this kind of the pre-opening before the real acceptance, you can show and exchange information, you think this is better to the community, or what is the basic feeling?
I believe it's a missing part in our community, and it is an important piece. Yeah, because the finest thing that the peer review system can do is really to question whether conclusions are right. Yeah, and but the peer review system sometimes really limits new ideas and say the data are not consistent and others have described this. Yeah, and if you publish it in a manner that everyone can see it, and we need of course a culture to discuss these preprint publications. So we had, for example, a renowned German virologist who published his paper in a preprint, and he was criticized in the media, that the statistics that he had in his publications were not correct. And he said, yes, it was not correct, and this is something we have to do. Yeah, it's the way how science works, that we do something, and someone comes up with a constructive criticism, and we improve. So that means we need the right culture, of dealing with this type of information. At the end of the day, I am a fan of Karl Popper, who said it is our task together to find out the truth. No one has the truth, but it is an approach and that emerges. The truth emerges when we work together.
Yeah, so I like this idea. The publication is not always the final answer. It's still in the process. Just one paper doesn't reveal everything. We have to accumulate many findings until we reach the final answer. So in that process, this preprint publication is one process. I totally agree.
Absolutely. This is so important. I really believe that the scientific community, so the accomplishment of making a vaccine in such a short time, it is an accomplishment of the whole scientific community. And I believe that the scientific community performed really excellent during this pandemic. And that this created also trust in the community, that science can make a difference.
But we have to tolerate some of this kind of preprint publication may not be correct. So we have to always be critical what we choose, what we don't take.
Yeah, absolutely. But that's the way if we have a healthy system, it will be able to find the truth.
In Japan, we have many pharmaceutical companies, and many of them also trying to raise a good vaccine for COVID-19, and the government also inject significant amount of money. And from outside, how do you feel the activities of the Japanese pharma?
To be honest, I'm not in the deep details of that, but I very much admire the Japanese way, of doing in developing new fields. I have seen that how, for example, the Japanese community dealt with immunotherapies. And the development of anti-PD-1 antibodies based on your discovery is the best example how powerful Japanese companies could take up new concepts and develop it. And I believe that the Japanese way of doing it is doing it in a particularly diligent way. And in a particularly coordinated way. It is a little bit different than the situation in the United States, where you have so many players and everyone is doing something fast, and then some company emerges, whereas others are failing, or most of the others are failing. And it's a different approach, and I believe it's a complementary way of doing things.
So how is in Germany? You worked mostly in Germany, but you had a good relationship with a big American pharma. That helped at least in a business way. That's very important. Also large scale clinical trial. And German scientific way of pursuing all this vaccine production linked with the big pharma. In a way, that's the ideal combination.
Yeah, I fully agree, Tasko. This is an ideal combination. The Germans tend to be perfect. They have a wonderful engineering expertise, and they have a wonderful education. But they are not anymore this big pharmaceutical companies in Germany. And therefore it's a great opportunity. For example, our partnership with Pfizer was extremely important and synergistic.
Good. Thank you. So maybe I kept asking the questions. You can ask questions.
Yes, I would love. So it's such a great privilege to talk to you. I'm absolutely a fan of your research and the way how you really continued not only to make a single discovery, but also continue to bring this discovery into application. So my question to you, it's a rare privilege for a researcher to make a major contribution to science that leads to an innovation that makes a real difference for patients. So I would like to learn about your personal experience. At what point did you first saw the clinical data showing that the therapeutic approach, anti-PD-1 blockade, worked? So that's the first part. When did you really realize that this could be a completely game changer for patients? What did you feel? And how do you think the field will further continue based on your insight?
Okay, so the history of PD-1, we found the molecule 92. And then we found the real function of this molecule around 97 to 8. And immediately we thought about the cancer can be a good target. And we worked and published the animal model experiment. That's 2002. Then we tried to develop good humanized antibody, and fortunately, I think this is again American venture capital jumped on, and they created good antibody. And I knew this group actually collaboration with Japanese pharma Ono, who got the license from us, and then actual clinical trial started 2006. And I often met these venture people in the meeting, and they were telling me it appeared to be working, but I never seen the clinical data. So I also started a clinical trial in Kyoto University Hospital. That 2011. And it's a very small scale, phase 2A type, 2 doses, 10-10. And doctor one day told me, patient completely, the tumor is completely gone. And that surprised me. And that actually, well, it's actually 2 out of 20. But that was something to me. And the most striking way I realized this going to be work is, of course, the first paper published by Susan Topalian in New England Journal of Medicine. That was actually 2012. But the most striking, which I really I'm convinced, is the publication from the European network, the randomized trial of melanoma. And that showed the complete blind test between chemo and the nivolumab, and showed a very large difference in the overall survival. And that convinced me this is going to work. And that data published at that time when our crown prince visited our university. And my friend showed the current emperor when he was crown prince. So he was very much impressed. And I also impressed. And that is the moment I felt this is going to definitely work and could be a game changer compared to the chemo. It's a
Yes.
real difference.
Wonderful.
Yeah. So did I answer all your questions?
Yeah, maybe what is your expectation? What will come
Future.
next?
Future.
Yeah, future.
Yeah.
Yes.
Yeah, both of us worked on the cancer, and I'm pretty sure you continue to work on the cancer vaccine.
Yeah.
And as you know, the current PD-1 blockade is not complete. Maybe roughly 30% patient respond. So we definitely have to improve this efficacy. And we are working on a different strategy. Basically, we try to combine some low, small molecules, which is not the chemo, but activate immune cells and make the more efficient way. So I'm pretty sure you keep making the better cancer vaccine, and many people try to combine with the PD-1 blockade plus, and I'm pretty sure you think about this vaccine plus checkpoint inhibitor combination therapy. So my belief and hope is all this kind of the combination trials by 1,000 people in the world will definitely improve the efficacy of cancer immunotherapy. And I'm almost convinced cancer immunotherapy is the goal of our conquer of the cancer, hopefully this century. Last century, basically most of the infectious diseases overcome by the antibiotics and combination with many hygiene, but hopefully we can overcome like your vaccine and many efforts combined to make the cancer not the really tragic diseases, but anyway, we don't live forever. So we want to live in peace, right? That's our goal anyway.
Absolutely, absolutely, absolutely.
Yeah.
Yeah. Thank you so much. This really answers my question. May I ask a second question?
Oh, certainly, Uga, please.
Yeah. So I heard that you advocate new concepts in science, and I also believe this is really important. So. Do you have some examples? Which new approaches do you see emerging right now? Which might help us to broaden our view on nature, on biology, on immunotherapy. Do you have some favorite examples of new concepts?
Well, new concept in cancer immunotherapy is certainly brought up by Burnett. That's 70s.
Yeah.
But myself, I don't think I had any conceptual breakthrough based on this immune system watching the cancer as non-self is a basic concept.
Yes.
But the immunological community didn't expect negative regulator as a molecule. And that was probably my contribution and Jim Allison's contribution. We showed here the molecule that regulates and that can be useful for making the immune system more powerful and attack the cancer. So I myself, concept in biology is slightly different from the physics or chemistry because it's so complicated, as you know. And we have to try many things. We cannot foresee something going to happen, but we try many things. And then someday, we got it. That's the style of the biology, I think. And the people call it later concept, but
Yes.
in reality, as you showed, we have to work hard in biology.
Yeah, absolutely. Absolutely.
Mm.
Yeah, yeah.
Mm.
So concept describe not the nature, but our way of thinking about nature, and we have to adapt. I fully agree. Biology is so complex that there is no rule which is forever. Yeah. It is always dependent on the context.
Yeah, one thing I'm now pushing is now people study one molecule or one cells deep, but often lose whole picture. So we learn the medicine, and we always taught we have to see the patient, not the disease. So that's we really need at this moment.
Yeah, thank you for saying that. It is completely right. So as a young physician, I had an experience. I did lab tests with my patient, and one of the lab tests showed that something is wrong, and I wanted to treat the patient, and my more senior physician asked me, "What are you doing here?" And I said, I'm treating this because this laboratory value is not right. And he said, but the patient is fine. So we are not treating laboratory values.
Right. Yeah. That's very important. Good lesson.
Yeah, that's
for
very
young
important.
doctor.
And this is what we have to consider. Yeah, it's exactly what you say, seeing the pieces but not the whole picture. Yeah.
So since you said that point, I'm pretty sure many people are now concerned or interested in the side effect or the how long the BioNTech vaccine contribute for prevention, and those kind of what is your perspective? You may have all these kind of data.
Yes, we are collecting the data, and what we see is we have two types of immune responses. This is the T cell response, particularly the CD8 T cell response, and the CD8 T cells will stay for a long time, and we believe that the CD8 T cells, together with the CD4 T cells, will prevent severe disease. Yeah, maybe for years. Yeah, and the good thing about T cells is everyone has different MHC, HLAs.
Right.
So the virus, it's difficult for the virus to do an escape. Yeah. But on the other side, we need antibodies to prevent infection. Yeah. And the antibody titers decline over time. We now see that after 6 or 8 months, we have a decline of the antibody titers. And on the other side, we see also the emergence of variants, which have antibody escape features.
Right.
So we believe that it would be useful to have a booster vaccination, sometimes between 6 to 9 months after the second dose. Right. And then we have to observe how long the boosted antibody titer goes. Yeah. And then based on data, we have to make decisions. So this is the part related to the booster. With regard to the safety, it is good that we are now getting more and more safety data. We have vaccinated more than 600 million people with our vaccine.
Wow.
Which really provides a way of sensitive detection of side effects. Yeah, and we are learning that there are certain side effects which might be rare, but higher than the background. We are communicating that we believe that our vaccine is a safe approach and has a excellent risk to benefit ratio. But we need to acknowledge that every drug has its side effects. Yeah?
Right.
So, and this is, and people need to understand it. We have to avoid that this is exaggerated, so people are too much scared. But we also need to avoid the impression that everything is harmless. Yeah, it is finding the right balance.
Yeah. So looks like we are going to finish, but I'm pretty sure you're going to open those new informations and keep us updated. And I hope we can see you in future someday, sometime, somewhere, and listen to your cancer vaccine. And we hope our progress will complement with your research. Thank you very much.
Thank you, Tasko. It's likewise. I hope that we will meet in one of the cancer conferences, and I would be happy to invite you to come to Mainz. We have a strong cancer immunity community, which admires your work, and it would be great if you could give us a plenary lecture.
Thank you, Ugar. It's a real pleasure. Nice chat with you, and further success in the various fields of your research, and also you're a businessman.
Thank you, Tasuku. We are all business people.
Right. Okay. Bye now.
Bye.